accomplished physician, population health scientist, epidemiologist, and biostatistician. He trained as a medical doctor at Lagos State University in Nigeria and holds 3 postgraduate degrees from Harvard University. Master's in Global Health Biostatistics, a PhD in Population Health Science. In between, he coordinated clinical trials and prospective cohort studies in Tanzania during a postdoc at Harvard. He also completed a postdoc at Brown University, working on studies set in the Philippines. His PhD thesis and much of his academic research are focused on clarifying the interaction of nutrition, infectious diseases, especially HIV and malaria, and maternal and child health. He co-founded and coordinates the Statsclic, a non-profit organization that delivers free web-based research methods training to healthcare professionals across African countries. Statsclic training programs have been attended by thousands of healthcare professionals worldwide. He also teaches epidemiology at Harvard University. He serves as the trial statistician on clinical trials led by the Center for Clinical Trials, Research and Implementation Science at the College of Medicine, University of Lagos. He is currently medical director at Genentech, Inc., a biotechnology company in California, USA. He leads evidence generation strategy and phase 4 randomized trials for an in-market drug for multiple sclerosis. Dr. Abioye is a highly accomplished researcher with more than 50 publications in high-impact journals. He has led and supported numerous research, including projects funded by the U.S. National Institute of Health, Gates Foundation, and the Biopharma. Ladies and gentlemen, a warm welcome for Dr. Ibraheem Abioye.
Thank you very much for the kind introduction. Thank you very much. First of all, protocols. Very grateful to CCTRIS for the invitation, the Deputy Provost of CMUL, the incoming Dean, all the professors, all protocols to be observed, medical students, residents. It's my great pleasure to be here today to discuss with you all this journey that clinical trials take from the time of conception to the time when we're delivering the evidence and essentially disseminating that evidence to the community. This is the outline of my talk. I will be talking to you about why clinical trials matter, what are the randomization and design considerations that we have. I will also talk about the process that the data from clinical trials take from the time of data entry all the way until the time when we're locking the database for analysis. I will talk about analysis planning and integrity. I will also give some real-world lessons. So you can see that this is likely to be a very heavy talk. And so what I'm going to do over the next hour or so is essentially introduce some key principles. And then if we have an opportunity afterward, we can discuss in some more detail. But I want to point out very quickly that my goal is to introduce concepts to you that will inspire you to ask more questions here, but also inspire you to do more research, to study further on your own, and I'm always available for questions at any point in time. As has been mentioned, I have been involved in a number of clinical studies here at CCTRIS, so a lot of the learning, a lot of what I'm going to be able to share today is actually learned from my work with CCTRIS, right? Every day that we do work, that we do research is an opportunity for us to learn. And so, learning never ends. So the why and how of clinical trials is the first part of my question. So what I'm hoping to be able to do, I think maybe if it works, will be to stop at certain points in time during this conversation, and if anyone has questions, we can take it. That's one option. The alternative will be to wait until the end and ask questions, and whichever one is most amenable to the team is best. Maybe the end is better, right? Okay, all right. So why do clinical trials matter, right? We all say that clinical trials are the gold standard for evidence. And if you think about clinical trials, in the hierarchy of evidence that we are all familiar with, clinical trials are usually at the top of that hierarchy. And part of what I hope to be able to, demonstrate to you is why clinical trials are actually at the top of that hierarchy. And how do we ensure that a clinical trial does not fall from that top hierarchy to a lower level on the hierarchy of evidence, right? So this is the goal of this talk. As you know, all of you who are here and who are listening probably have an idea of what a clinical trial is. Maybe you've also participated in one. But very often when we're thinking about clinical trials, we're talking about often randomized, not all the time, studies of interventions, and those interventions tend to be therapeutic interventions, right? So most often when we're thinking about clinical trials, we're thinking about them in the context of drug trials, and in the context of interventions that we think might be able to help modify our clinical practice. But those are not the only types of clinical trials that exist, right? So you can also study diagnostic interventions, right? To assess whether screening for HIV using one approach versus another approach, to what extent does that influence care towards as the person then goes along in their clinical journey, right? You can also have not just diagnostic, but also preventive intervention. So vaccines, for instance, are one that we have all, if you've lived recently through COVID-19, vaccine trials took center stage, and thank God for vaccine trials, right? So these are all opportunities for us to conduct these studies. And the key point in these clinical studies is this. So what we're always trying to do in clinical trials is there's an eligible patient sample, right, of all the people who we could study, right? If you think about it, you're trying to study, and an example will be pregnant women in Nigeria. That is maybe 1 million people at any point in time. And there's no way you can study 1 million people in any clinical trial. First of all, you will not have enough money to do it. It will be logistically challenging. And it's not necessary, right? So what we always do is I will take a sample of that eligible patient pool, right? And then we follow up over time. So we intervene on this sample, and then we follow up over time. So time is a key factor in clinical trials. This is always important to remember. But what are we really trying to do in clinical trials? We're trying to find the truth. We're trying to find the truth. So truth is really, really central. So when we come to work on a clinical trial, we have to come with no preconceived notions, right? So you have a hypothesis that you're trying to test, and you believe that hypothesis, potentially you might say, if I provide iron supplements to pregnant women, that it will improve their risk of anemia, right? But you want to come to your clinical study bearing in mind that there's a potential that, in fact, iron supplements might not work. Okay, and you should not be trying to tilt the evidence in any direction whatsoever. Right, so a lot of the things that we'll talk about over the next few minutes will relate to how do we ensure that the clinical trial does not end up being modified inadvertently by the people who are conducting the studies. Okay, so that's always one of the goals that we're trying to achieve. So there are two key words that I wanted to walk out of this room today knowing what they mean. And one of those words is precision, the other word is bias. Okay, one of those words is precision, the other word is bias. So when you're conducting any research study whatsoever, there is the concept of randomness that happens in your assessment. Okay, so if you have two clinicians assess the same patient, they might end up with different diagnoses. If it's such a clear condition, maybe they have slight differences in the diagnosis and they have a general truth that they are both reached. Okay, so that those slight differences that they're coming to, that's where precision comes into play. And when you propagate that over hundreds of people, then it begins to widen to some degree. Okay, so another way to think about this is if someone were to ask you what is the prevalence of HIV in Nigeria, right? If we went around this room and asked everyone what the prevalence of HIV in Nigeria is, and maybe one person says 3%, another person says 5%, another person says 8%, right? We might, assuming that we're all fairly knowledgeable about HIV, we will likely come up with a range of values. Okay, the extent to which that range of values is narrow, that is precision. That means that although we are estimating with some error, right, none of us, maybe most of us are not actually correct, but we're very close. So that precision is one of the key factors that we're trying to achieve in clinical trials. So we're not assuming that clinical trials will always perfectly answer the question, but we appreciate that there's likely to be a little bit of a range, and that's okay. But we want that range to be as narrow as possible. But the other key concept is bias. And bias results when a clinical study, say, let's go back to this HIV prevalence that we were saying. If, for instance, the true prevalence of HIV is 4%, and for some reason, when people are asked what the prevalence is, they keep saying 20%, right? That there's likely to be 4% and 20% are meaningfully different, right? And there's likely to be a reason why they're getting it wrong. And that reason why they're getting it wrong is likely to not be due to precision, because it's so far away from the truth. Is more likely due to be due to bias. Another way to think about this is if I asked you if iron supplements are likely to affect the risk of anemia, okay, and I keep using iron supplements a lot because that's a lot of the research that I have done, but also that's some of the research that CCTRIS does. Okay, so if iron supplements, if someone asks you if iron supplements are effective, and you see that iron supplements are not effective, despite overwhelming evidence that they are, then there's likely to be a reason why you're finding that different result. So bias, what bias does is that it alters the finding of your result. It might change the entire direction of the finding of your result. And so when bias happens, that's a huge challenge. And that's one of the key factors that we're trying to prevent. So we want for there to not be bias, and we want to minimize randomness. We want to increase the precision of our study. So this kind of sets the stage for the rest of this discussion. So many of us might be familiar with this classification. So when we're thinking about clinical trials, we might describe them in terms of the phases of development. We might say phase 1, phase 2, and you know, when Dr. Bala was introduced, it was said that she's the phase 3 lead of the clinical trials, and Dr. Okmade is phase 1 and 2, right? So those phases, they mean something, and they actually correspond to different points in time. Traditionally, when we think about development of a new drug product, okay, or a new device, right? In academia, we don't always use that classification. Sometimes we're more likely to use descriptive terminologies. You might say something like, this is a dose finding study. And a dose finding study is more than likely to be a phase 2 study. Right? We might say something like, this is a biomarker study or a translational study, where there is evidence from the studies that were conducted in animals or in cell lines, and then we're bringing that evidence to human populations. So that's translational. So the key point that I'm trying to make is that there are multiple ways that people will describe clinical trials, and it's helpful to know that these different definitions ultimately are trying to describe the same thing in different ways. Okay? Clinical trials follow the rigorous scientific method, which we're all familiar with. First of all, there has to be, we have to define the research question in terms of what we call a PICO framework. And many of us might be aware of what the PICO framework is. Every research question that is conducted in humans, you have to be able to identify what the exact population is, right? What the intervention is, what the comparator is, if there is a comparator, and there isn't always a comparator, and what the outcome is in that clinical study. Okay? And once you then define that PICO framework, the thing you want to do is define what your target experiment is, or often also called a target trial. A target trial is the ideal trial that you would like to conduct if you had all the money in the world and you had no ethical constraints. Okay? If you had all the resources available to you and you had no ethical constraints, what trial would you conduct? What is the perfect trial to answer this question? Okay? But the reality is that we cannot always conduct a target trial, right? Because these are human beings. We cannot subject them to the interventions for as long as we want, in whichever way we want. And so we have to essentially go from that target trial and come to what is actually the kind of experiment that we can conduct. Okay? But it's always helpful to have that target trial in our minds. So that difference between that target and the experiment that we actually conduct is where the errors usually come from. Ultimately, what we're hoping to be able to do is to ask a clinically meaningful question, right? Answer it in a way that is convincing, and while we're doing that, ensure that we're not harming patients in the process. So why does randomization, so every time we think about clinical trials, a lot of times, most of us are thinking about phase 3 clinical trials where these studies tend to be randomized and controlled. Okay, so if you don't leave here today with any other understanding, I would like for you to know why randomization works. Okay, at the point when a clinical trial is randomized, at the point when people are randomized into a clinical trial, that clinical trial is unbiased, right? Bias happens after randomization. Okay, and this is a general principle for you to take away with you. Therefore, randomization is very critical. But how does randomization work? Why does it work? If you were to, and I like that example that Prof gave of the Babylon, and so if you think about the ideal experiment, right? When a patient walks into your office and you're trying to define whether maybe an antihypertensive that you're providing to them, whether it effectively reduces their blood pressure. Right. So what you would want to do is take that one patient as they walk in, you duplicate the patient. I don't know how you will do that. Some machine, pass them through a machine, right? Give one version of the patient a drug, and then don't give the other version of the patient. And then after some time, look at the blood pressure of that patient. But we cannot duplicate human beings, right? So we need a different way, and that's why we will therefore do it on a larger scale. So we will bring multiple people, right? Multiple people into the room and run and divide those group of patients into two. Okay? So instead of one person, therefore, we will have maybe 100 people, maybe 1,000 people, and we will divide them into two groups that are identical. Okay? Those two groups, we want those groups to be so similar that it's almost like, thank you. So we want those two groups to be so similar that it's almost like they're mirror images of each other. And that similarity will be on the basis of certain covariates, certain factors like their age, the distribution of the sex, whatever other factors, depending on the population of interest and the clinical characteristics. So we want those two groups to be equal and similar, such that the only difference between them is that one group gets the treatment and the other group does not. That similarity in the groups is what we call exchangeability. We want those groups to be exchangeable to each other so that it doesn't matter whether you give group A the drug or you give group B the drug. It doesn't matter. But what matters is that the group that you do give the drug to, you can compare their outcomes after some time to those you did not give the drug to. Okay? This is the fundamental concept of randomization, and this is why randomization works. I've talked about the differences between an ideal experiment and clinical trials, and there's always going to be factors that you cannot control in clinical trials. It might be that maybe because people, people are people, right? Maybe they don't. You ask them to take their supplements every day, they don't, right? Maybe out of 100 days, they're only taking it for 70 days, right? Or they come to you one day and say, I'm no longer interested in this study, I would like to withdraw. Right? These are all factors that make the clinical trial to not be an ideal experiment. Always, we want the clinical trial to be conducted ethically, right? We want people to provide informed consent, right? That means that they have to understand the intervention, they have to freely approve their participation, and they have to understand that they can withdraw at any time, and they should be able to withdraw, right? So these are all factors that we must account for and allow for in a clinical trial for it to be ethical. We want to make sure that the clinical trial that the study or the intervention is not introducing undue harm. Okay? But there's another thing that we want to ensure, and that is respect for the persons. And that is that we want to ensure that the people who are participating in the trial are also those who are most likely going to benefit from the treatment when the trial comes, when the trial findings get into the real world. So if you're conducting a study that is among, say, people who you're looking at physical activity in the community, and then you then go conduct that study in a prison, right? That means that essentially you are exploiting these vulnerable people who are unlikely to benefit from the intervention because they're incarcerated, right? So that's something to bear in mind. In clinical trial regulation, there's multiple ways that clinical trials are regulated, and the one that most of us are familiar with are ethical committees, right, and IRBs. And the NHREC is National IRB for Nigeria, and very important. It's great that we have that facility, and there's ethical committees at universities and teaching hospitals all over the country. Okay, but there's also good clinical practice, and in the International Council on Harmonization, there's always certain guidelines for clinical studies to be conducted. We also want trials to be registered, and the reason why registration is important is so that there is limited manipulation, right? You want people to pre-specify the protocol and have that online so that they don't, so that they when they do make modifications, they are compelled to also report the modifications that they make, and we can fully look at the life cycle of the trial and understand where changes happen along the way and why those changes happen. It can help us to have confidence in the results that we then come across from the study. Okay. All right. For the next few minutes, I'll introduce you to a number of concepts about how clinical trials are designed and some of the steps that, some of the points at which key decisions have to be made in clinical trials to ensure that the evidence from them is rigorous. The protocol is the first one. I've talked about the PICO framework, which is population, interventions, the comparator, and the outcomes. So we've talked about that. So sample size is another one. You want to ensure that the clinical trial has a large enough sample size. We also want to ensure that the data collection forms that you're using, the case report forms, are done in such a way that you're minimizing missingness from your studies. You want to ensure that the way that the forms are designed, you want to ensure that people are not probed to ask questions in such a way that the answer is predetermined. Okay, so there has to be neutrality in the way that the questions are framed. Data cleaning is really central to clinical trials. You want to ensure that the data is that the data values are reasonable, that the dates, and this is one of the things that you come across when you're conducting clinical trials, is the challenge with dates. Okay, the first one that you will likely encounter is when you go from December to January and people are still inputting dates from December, right? And the year is completely out of whack, right? But also, people make mistakes. There's the fact that some clinical trials are using the month, day, and year if they are US studies, while some others are using the day, month, and year. And these are key differences that when you're entering the data and you're reading the data back, you need to understand how the dates are formatted, right? Very small, but it can completely change the findings from your study. Okay. And then once data is clean and you lock the data, before you lock the data, you want to finalize the statistical analysis plan. And we'll talk about that again as we go along. There are some common clinical trial designs, and this is the most common design that many of us are familiar with. There's two arms, and one group of patients gets the experimental intervention, and the other group of patients get placebo, right? One modification of that is instead of placebo, they might get the standard of care, right? And this is the basic design for clinical trials. But this is not the only design, and it really depends on what the goal of your clinical trial is. There are some times when we're trying to test the effect of timing. So we might have some group of people start on one intervention, and then maybe after some time, they will then switch to the experimental arm, right? So we can compare the effect of whether they started early or they started late on the finding. So maybe one way to operationalize that is when you have HIV patients diagnosed, right? And you realize that every day counts. And we know that every day counts in HIV care because studies like this were conducted, right? Where you can look at the difference between starting antiretroviral therapy at the time of diagnosis versus starting it later. And there are other designs as well. We can test combination therapy. And combination therapies are really important because sometimes when you give two drugs, the effect of those two drugs is not necessarily an addition of the effect of one and two, right? So sometimes there's interaction, and that's why it's helpful to assess combination therapies of drugs. Sometimes we're testing switching, whether people switch from one standard to the next. There are also crossover designs, and these are also important when we are trying to look at the effect of one drug versus another. And sometimes we want to see maybe in practice people are likely to move from one treatment to the next. The key challenge with crossover designs is carryover effects, right? So if you provide the intervention now, so most drugs have a long duration of time that they're working for. So if you have a drug that maybe its effect carries actually until like 6 weeks, right? Then if you're administering the next one before that effect has waned, so let's say after 1 month, that means there's 2 weeks during at which the drug that you first of all provided is still being effective, right? So if you conduct a crossover study, you have to have a washout period at which that carryover effect is then going to be nulled. Okay. There are other designs as well, and factorial designs help us to study, instead of conducting two experiments, you will conduct the same two experiments on the same group of people. So instead of, first of all, doing one trial now, an example of a study that I was involved in Tanzania, we were looking at the effect of vitamin A and zinc supplementation on the risk of placental malaria, right? So these are two interventions that ordinarily you would have compared each one to placebo. So instead, what we then did was that there were 2,500 women, and we divided them into four groups. Okay. In the first group, you provide vitamin A. In the second group, you provide zinc. In the third group, you provide vitamin A and zinc. And then the fourth group, you provide placebo. Right? This is essentially two experiments in one, and you can compare the findings of this study as if you have two experiments. So it helps to save money, it helps to save time, and there's the statistical power as well. That's a key benefit of that. And sometimes, like we have in ASNV today, you want to randomize the cluster. Okay, and these are cluster randomized trials. And when we say the cluster in this case, so very often in clinical trials, we are randomizing the individual. If there are 2,500 people, each person has a chance to be randomly assigned to one treatment or the other. But in a cluster randomized trial, we would randomly assign maybe the clinic or the community. We might say that if we had like a cluster randomized trial of the teaching hospitals in Lagos or the secondary level hospitals in Lagos, one general hospital will get treatment A, one general hospital will get treatment B, and the treatment that that general hospital is assigned to, all the patients in that hospital will get that treatment. So the advantage of a cluster randomized trial is that it limits contamination. You see, one of the issues with clinical trials is that when you provide a treatment to one individual, right, and that person is sitting in clinic. So you know how women, when they come in the morning to antenatal care, they're chatting, right? They're saying, oh, I'm in this study, this is what they gave me, it looks pink. And the other person says, well, I'm in this study, this is what they gave me, it looks yellow. And one person is saying, oh, on this drug, these are the effects that I'm observing. And so this other person, oh, I think I know that drug, it is iron or it is multivitamin supplement. And so the person that you did not provide the multivitamin supplement to goes outside to the pharmacy and procures it. That is contamination, right? Another level where that might happen is when and maybe I should wait and tell you this example later, but contamination is a key challenge in clinical trials where, in some clinical trials, and if you want to prevent that, you can use cluster randomized designs. The challenge with cluster randomized designs is that you need a large sample size, right? So instead of maybe 2,000, you might need 4,000 people. And, that means logistical challenges, that means more resources. And when you randomize, you want to conceal the allocation sequence, right? Until the individual who has to know, some people will have to know the allocation sequence, and that tends to be maybe a pharmacist who is providing drug or whoever it is that is actually providing the intervention, right? So you want to conceal that allocation sequence until you have to disclose it, right? Because the challenge is that people sometimes, like I was saying, people sometimes will select to participate in your study based on their knowledge of the allocation sequence, or they might choose their arm. And how is this likely to happen? Say the doctor who is taking care of the patient or the research nurse knows that the next person who is going to come in is going to get treatment A. And so in order to prevent their friend or their sister who comes in because they want their sister to get treatment B, they will say, well, why don't you wait? Let the next person come in, right? So you ultimately have a scenario where if a research nurse or a research staff is aware of the allocation sequence, they might, without, we're human beings, right? So they might then be tinkering with who gets what. And if they tinker with who gets what like that, then bias is going to result because your population might not be balanced in the way that you want them to be. So there are ways that we will conceal allocation sequence. One of those is to have an independent statistician generate a sequence, or to use something like sealed envelope, which is what we use here at CCTRIS, to consider allocation sequences that are not easy to predict, or to allocate sequences across trial sites. So these are some strategies that are often used to conceal the sequence, but most importantly, do not reveal the sequence unless people must know. Okay? So I'll skip this because I think it's a bit, but the key point here is that there are different approaches that we're using to generate allocation sequence, and sometimes we're doing a very simple randomization, but other times we're using blocked randomization, and we can discuss it some more if necessary. Clinical trials have a primary objective that you're trying to achieve, and that primary objective is always the one that corresponds to the primary endpoint. When people are designing clinical studies, they list all these primary objectives, but they don't tie it very nicely to the primary endpoint, and that's a challenge, that's a flaw. So we want the primary objective to be tied to the primary endpoint. And an example of that is if you have a new drug that, you're saying that you're studying whether the drug reduces 30-day mortality compared to placebo, then the primary endpoint in that case should be 30-day mortality. So you will tie the primary endpoint to the primary objective, and the sample size that you estimate for your clinical trial has to be based on that primary endpoint. Okay? And if you have more than one primary objective, because sometimes you do have to have more than one primary objective, then you want to adjust for multiplicity, and you can talk to the statistician. And this is another opportunity for clinical trials to introduce, for bias to be introduced, is in the selection of the endpoints. Wherever possible, you want to select what we call hard endpoints. So hard endpoints are basically objective endpoints that if two people, there's always very likely going to be agreement on what this endpoint is. It's either a person is dead or they're not dead. Right? And death is the ultimate hard endpoint. So if you have a study and you're trying to choose between what the endpoint should be, that will be the primary objective of the study, then hard endpoints are always better. Soft endpoints are those endpoints where there is some subjectivity in the assessment. Okay? And every time you have subjectivity in the assessment, it means that observer judgment becomes an issue. And two people might examine the same patient and come to different diagnoses, right? This is something that we are all familiar with. And as a result of that, your clinical study might lead to different studies depending on who is doing the assessment. So hard endpoints are always better. And if you have soft endpoints, because sometimes you can't not have soft endpoints, then blinding becomes very critical. So what is blinding? Okay, so the idea of blinding, and a lot of these terms that I'm talking about, if you're already familiar with clinical studies, they're probably the bread and butter, right? Blinding is something that if you work in clinical trials on one day, you will be familiar with what blinding is. And the idea of blinding is that at least one of the key stakeholders does not know what intervention the participant received. Okay, that's blinding. And the level at which the blinding happens can differ. It might be the level of the patient, which is single blinding, or it might be that the patient and the healthcare worker that is taking care of that patient, both of them might not be aware of what intervention the patient received. But there's also another level of outcome of blinding, which is the triple blinding, where the outcome assessor is blinded. And I'll give you an example. If, for instance, the outcome of interest is AIDS-related mortality, and AIDS-related mortality is an assessment where a patient has died and someone then goes to the patient's records and then makes a determination that this HIV patient who died, did that patient die because they had AIDS? Because they had AIDS-related mortality. So this is a clinical judgment, but this is an assessment that the person who is conducting that assessment does not need to know whether the patient received treatment A or received treatment B, right? And because they don't know what treatment the individual received, it is not likely that it will influence the extent to which they classify this person as having had the outcome or not. Okay, so this is the thing, and biomarkers are one way. Another way, so if you collect blood samples, then the person who is testing blood sample does not need to know what treatment the patient received, right? So every time you're thinking about endpoints, you want them to be objective, but you also, to the extent possible, you want to blind. And even in open label studies, and we're conducting open label studies now at CCTRIS, right? Even in open label studies, you want the outcome assessor to be blinded to the treatment that the person received. The person in the lab does not need to know if this person received IV versus oral iron, right? Does not need to know what treatment they received. And this is something that we're ensuring at the center. Okay. All right, so let's change gears a little bit and talk about data management. But the first thing that I'm going to talk about is baseline assessments. If you have read clinical trial papers, you will come across in table 1, very often, there is one table that will summarize the characteristics of the patient, okay, by treatment arm, right? The characteristics at baseline by treatment arm. And the reason why we are doing this is, first of all, to demonstrate to the reader that these individuals in the study actually meet the eligibility criteria. But another reason why we're doing this is to assess whether the treatment groups are comparable. Okay, if the treatment groups are comparable, it gives us some confidence that exchangeability was met. And if you remember, I said that exchangeability is really central to clinical trial rigor. Sometimes when we're evaluating change over time as well, we want to assess whether we want to have the assessment at baseline, so we'll use it as a basis to examine change. And the key question that people who are handling the data in the clinical study are always worried about is the sources of error. The sources of error in data collection. Now, there's multiple potential sources of error in clinical trials, but potentially the most common one is just that the data is simply missing. Okay? And missing data is a huge, huge problem. And we'll talk about it extensively over the next 30 minutes or so while this talk is wrapping up. So I will skip it for now. But very often when people are thinking about error in clinical trials, they're thinking about simply that the data was fabricated. You know, in my experience, clinical trial data is hardly ever fabricated, but it's not impossible. So the investigators that are designing the clinical trial have to ensure that there are ways to check the data from time to time, so that if there is someone who is tired and just entering some random stuff into the database, there are ways to check and look out for those patterns. Okay. Large variability is another potential source of error. And then the final one is long delays in submission. So if you have a scenario, I'll give you an example where the research staff called the patient and found out from the patient whether the patient experienced a specific endpoint or not. It might be, let's say, pain. Okay, and the patient said, yes, I experienced pain and it's of this level. And usually you will grade pain from maybe 1 to 5. And the patient says, oh, the level of pain that I experienced was 4. Now, the research nurse then gets a call from their friend and they start yapping away for 30 minutes. They have not entered the data from that patient, right? The fact that they didn't enter the data from that patient means that when they then come back to enter the data from that patient, they might not remember if that patient said 4 or they said 3. Right? This is one potential error. And this one is fairly more common, right? That's why one of the ways that you will have to address that is through training and making sure that the participants, the staff in the research are well trained to prevent scenarios like that. Missing data happens very often because participants withdraw from the study or because they're lost to follow-up or drop out. Okay? Sometimes people just miss visits, and missed visits happen very often, right? For whatever reason, they cannot come now, or sometimes the data is just lost. Today we have cloud, right? And that means that a lot of our information is backed up all the time. But in the olden days, where you will have to carry maybe a CD-ROM from place to place, and you're the investigator, and then you forgot it somewhere, that means that all that data from the clinical trial is gone. Okay? Or it might be specific, variable, specific CRFs from that study that are just lost. Sometimes the item is just missing. How do you prevent missing data? What do you do about missing data? You really just can't prevent it. And the way that you prevent it is in the design of your study, in the way, so very often in clinical studies, we will have very rigorous follow-up reminders. So when I started working in clinical trials, I used to be very uncomfortable with the way that we will call, and this was in a study among pregnant women, and we will call these women as closely as possible. I imagine that maybe we're the ones calling them the most, right? And the way that we will call is as pregnancy is advancing, the frequency of the calls are increasing, right? Because we're trying to target that, make sure we were collecting placentas at delivery. So you can imagine that we always had to be present at the time when the woman is delivering. So we were calling them on an almost daily basis in the week when we expect that their EDD is, so that they're the ones that are top of their mind when they remember, oh, I have a baby, this is my husband, I am in this clinical trial, right? It has to be like that top of mind for them. So once they start experiencing the pain, they're calling you. That's the only way that you're not missing that data. So over time, I realized that if you actually skip on these calls, you're going to miss that data, right? So reminders, follow-up reminders are really critical in clinical studies. But also today we have REDCap, and a lot of data collection is online and is electronic data capture, and that is such a huge opportunity, right? Just 10 years ago, you will have to collect the data on paper, and then you will have people enter that data onto the computer, right? And what that meant is that between the time when the data is going from the paper to the computer, a lot of things can happen, right? There's errors in entry that can happen, and you have to then go back to verify the data from your source. And if data is missing during the time of your collection, you have an opportunity to go back and fill that data, the gaps that exist. And this is really, really important. As you're advancing in the conduct of the clinical trial, you want to go back, you want to assess how much missingness you have, right, before it becomes too far out. And this is most critical for the study. So you have to prioritize, these are the endpoints, or these are data points that you want to track the closest, because you cannot track everything, okay? And for the ones that you choose to track, you want to make sure that you're going back to the mom and you're figuring out exactly what happened in this case, or you're going back to the study participant, as the case may be, and you're figuring out exactly what happened. And if you work in clinical trials, you probably are familiar with the concept of windows, right? The reason why we have windows is so that we can limit variability in the clinical trials, right? And let me explain what I mean for people who might not be familiar with what I'm talking about. When you follow up a woman who has just delivered, and you want to assess that woman at 6 weeks postpartum, right? In reality, you'll be able to assess many people at 6 weeks postpartum, but it's not everyone that you'll get to assess. For some people, you might get to assess them a little bit after 6 weeks or a little bit before 6 weeks. And that's okay. Earlier we talked about precision, and the range should be limited. But now imagine that instead of at 6 weeks, this person is showing up at 13 weeks for a 6 weeks visit. Then it's unlikely that the biology has completely changed for a person. It's almost like the body does not recognize them as having been recently pregnant, right? So that means that you have to have a window during which you say, okay, even if you cannot come at 6 weeks, can you come a week before or come a week after? Oh, I'm going to travel for work, blah, blah, blah. Can I come 3 days before my visit? And say, yes, let's make that happen. Or, okay, you missed that visit, and you're calling and say, can you come tomorrow? right? So that window is really, really critical to limit variability in the data that we are collecting, okay? Another key challenge is the use of non-standard terms. In a clinical trial, we will usually define the terms that we want to use, and we would hope that everyone who works on that clinical trial understands the definitions that we're using. So in SNV, for instance, right now, we've defined the definition for sepsis. Okay? Another clinician might have some other conception in their mind from wherever for how they expect that sepsis should be diagnosed. So if they are working on the trial and they don't discard that definition that they have in their heads, they're likely to cause problems for us. And these are the kind of things that you will look for at the time of training, and making sure that the research staff actually anticipate that challenge and prevent it. The other key point that I want to take away from this talk, earlier I said I talked about precision and bias as being two terms that if you walk away from here, you should hopefully remember what it is. And this is the other point that I want you to take away. And that is the concept of random error and systematic error, okay? When error is random meaning that there is no way to predict what the error is, right? While systematic meaning that you can guess what that error is, you can predict that error based on some characteristics that you've observed. Okay, and so a way to operationalize this and to help you understand it better is, for instance, when you measure blood pressure and you give two people the same person, two research staff are measuring blood pressure on the same person, they're likely to place the cuffs in different ways. They're likely to listen to the blood pressure in different ways and ultimately provide different values, okay? That error that happens is very likely to be random. There is no way to predict how and why the blood pressure reading is that way, okay? But one example that you might have is, say for instance you are only assessing this outcome in one arm, you're not assessing it in the other arm. Or let's say you're assessing in one way in one arm and you're not assessing it in the other arm. So that difference in the way that you're doing the assessment in arm A versus arm B is a systematic error. Okay? And I'll give you an example. If you have a clinical study where you're treating patients for cancer, okay? And I'm just generating this example as I go. Now, you're treating patients for cancer, and after like 6 months on treatment, the treatment is so effective that one group of patients, the ones who received the treatment, they are visibly better than the ones who did not receive the treatment, okay? When the two groups of patients come into the doctor's clinic and the doctor is having to ask them about, say, pain or loss of function, it is more likely that the doctor will ask more carefully in one group versus the other, because the doctor can visibly see that maybe one group of patients are always walking in, straight, while one group are always walking in on wheelchair. It's more likely that they will be asking about pain in the ones that are coming in on the wheelchair compared to the ones that are coming in walking, right? And it's just human. It's not something that they intended to happen. But that difference in treatment in the group A and group B is a form of systematic error, okay? And that's important to anticipate. That is important to anticipate in people who are, in participants of clinical studies. And so sometimes we will have audits that help us to track some of these errors, and it might be like a structured audit, or it might be routine where this is random sample, 10% of people are going and you look in the detailed information of that 5 to 10% of people, or you're verifying the data for specific variables in a small number of variables. One of the key issues that you will encounter in clinical trials is the DSMB. And the DSMB is a data safety monitoring board. And what the DSMB does is that they look at the data in the clinical trial from time to time. This is an independent board, not the investigators who are working on the study. And they will look at they will look at the data in the clinical trial and then decide whether the clinical trial should continue or whether it should stop. Okay? They determine whether it should continue or whether it should stop. And very often they're using some decision criteria to determine whether the clinical trial should continue or stop. And some of those are harm, right? If there's evidence of harm in one study arm, right? There's harm in one of the groups and the other group does not have that. And maybe the group where you're seeing harm are those that are actually receiving the intervention, then you know that maybe it's time to stop. Okay? Or there's given an expected benefit. And the reason why the DSMB basically exists is to protect the patient from two things. So it's either that the patient is being hurt by the trial, or the patient is experiencing overwhelming benefits from the study such that we can see that this is a drug that works, we should stop spending money and we should start using it in the clinic. Okay, so that overwhelming benefit that is being experienced, once we see that, it's time to stop the trial and then just go, right? So the one reason why we want to do that is there's ethics, but there's also costs. We don't want to keep spending money on a drug that you already know works. Right? So these are the two key reasons, and everything else relates to that. So if the quality of the data in the trial is limited and poor, that's another reason why you want to discontinue, because you're wondering if these people cannot put together good quality data, maybe there's no point continuing to spend money on that study. So I'll talk a little bit about analysis planning and the steps that we take to keep the integrity of the trial. The first is that the statistical analysis plan, right? As the clinical trial starts from the protocol stage, you want to put together a statistical analysis plan. And that statistical analysis plan should anticipate all the decisions that you will need to make in the analysis of the clinical trial, right? So usually when we will put that together here at CCTRIS, we will, share with other statisticians. They will take another look. We will, share with the team, and people will send comments. So it's not something that you do in a silo, because usually you will need to share that statistical analysis plan with the paper that eventually gets published. So you can think about it's a public-facing document, right? And when you're submitting the paper, for instance, sometimes you're required to submit the statistical analysis plan. Sometimes you're required to share the data from the trial. Sometimes you're also required to share your code, okay? So somebody else should be able to look at the entire process and come to the same analysis findings using the code that you make available. Also, they have to be able to replicate the work. Even if you don't make the code available, they have to be able to read your analysis plan and say, okay, this is what they did, now let me do it. And then when they do it, they should get the same results that you got. Okay, so this is very important to bear in mind, that the statistical analysis plan has to be finalized, and it has to be done before unblinding the data set, before we know what treatment each subject received in a study that is a blinded study, obviously. Okay. And one of the key questions, I'll talk about a few quick key questions that we are always asking in the analysis of clinical trials. And the first one is, what population should we be analyzing, right? And there's three common populations that we're always thinking about in clinical trials. And the first is intention to treat population. An intention to treat population, these are the people who were randomly assigned. If you remember, I said that at the time when you randomly assign participants into treatment A versus treatment B, that your clinical trial is not biased at that point, and bias happens afterward. Okay, regardless of whether the person uses the drug or does not, regardless of whether they adhere properly to the treatment or not, regardless of whether they withdraw from the treatment later or not, you're going to assess them assuming that they actually remained in your study. At that point, that analysis, that population is an unbiased population, and that intention to treat population is the one that you will prioritize. But there are other populations, and sometimes people will say, oh, we want to look at the people who actually use the drug correctly, because we want to be sure that this group of people actually experience the outcome truly. So the intention to treat population will represent the real world, where some people use the drug or don't, some people withdraw or stop using the drug ultimately, some people don't use it properly, okay? That's the real world scenario. That's how we use medicine in the real world, okay? But in the power protocol analysis or in these other forms of analysis, we're making modifications to the analysis population to be able to accommodate and to ensure that we're looking at a subset of people who hopefully actually benefited from the treatment. Okay? And so this is a subset, always a subset of the ITT population. So the modified ITT population is a subset of the ITT, and sometimes the power protocol analysis population and the modified ITT population are similar, sometimes they're not. So it all really depends on how the investigators define these terms while they're putting together the SAP. Okay? We want these three populations to be as close as possible. We don't want all this white space. Okay, we want this to overlap as much as possible. And the ideal thing to do is to define these three populations before you do the analysis, and when you do the analysis, to compare the findings. If the findings from your ITT analysis is different from the others, then you have to be concerned that maybe your clinical trial was biased. And I'll show an example as we go along. Protocol deviations are just essentially when a person who is supposed to participate in the trial in some way do not participate in the trial in the right manner that they should. So maybe they withdrew, or maybe they deviated from the study, and we talked about that, where the person might switch, or maybe the eligibility, they were not really eligible for your study. And sometimes when you're trying to do your analysis, you will take out those people who deviated, and that is the power protocol analysis, right? People who experience a major form of deviation. And it might be that, so when we talk about a major form of deviation, what we're thinking about is that deviation that is likely to influence the outcome of the study. Okay, so sometimes when we take these people out, first of all, we want it to be a small group of people. But if you do have major deviations like that, then you will analyze the population differently and then compare your findings in both groups. So that's protocol deviation. These are key questions that you want to ask. If you have more than 5% of protocol deviations in your clinical trial, nobody's going to believe the findings from your study. Okay, so this is something to always bear in mind. We want to train the staff as well as possible in the beginning and throughout the study to prevent occurrences like that. Another key question that people are always asking is, what data from the clinical trial should you report? And as you're thinking about this question, the thing I want to tell you is report all of the data. Okay? Report all of the data that you have, all the time points that you measured, all the subscales in an instrument, all the definitions of the outcomes wherever possible. Unless you have pre-specified in your statistical analysis plan that this is the definition we're going to use. Okay? If you did not pre-specify in your SAP, and you're coming to that decision later, then you have to report everything. That way, people can see that you're not selecting based on the findings, right? We want to see that you're not trying to just show us the best parts, okay? We want to see the full picture of the analysis. If you analyze and you have unadjusted and adjusted findings, you should report both of them. If you have a composite endpoint, and we haven't talked about this today, but sometimes in clinical trials, instead of having one single endpoint, we will have multiple endpoints and we will combine them together as a composite endpoint. So if you have a composite endpoint, and maybe an example of that is you might say that we're going to look at disease progression and death, and that's a composite endpoint, right? Because disease progression is one endpoint, death is another endpoint. And if you now merge them together into a composite endpoint, you have to report the findings for the composite endpoint, the findings from the components of that composite. So in this case, you have to report the findings for disease progression and death, the findings for disease progression, and the findings for death. Composite endpoints are an interesting part of clinical trials, and the challenges that are numerous, but the key takeaway from this session today is that you have to be as transparent as possible with the composite endpoints if you do use them. Missing outcome data can be concerning. I've talked about missingness multiple times, but the key takeaway that I think I want to take away from this conversation today is missingness becomes an issue in two ways. It becomes an important issue, a very important issue if two things happen. If the missingness is huge, say maybe more than 5%, and the worst scenario more than 20% of your data, of your outcome data is missing, and that's a key challenge. Or if the missingness is more in one arm versus in the other arm. So it might be, for instance, and this happens, these are not hypothetical scenarios, these happen in clinical trials. You have a situation where, for whatever reason, the people in one arm of the study are withdrawing. Okay, they might be withdrawing because of adverse events. So maybe the intervention causes a lot of nausea and vomiting, and they're like, oh, I'm not going to participate in this anymore. And very soon they start calling their phone and they stop picking. Okay, so if you have all of that happening in one arm of the study and it leads to withdrawals and it leads to missing outcome data, that's a huge challenge that you have. Okay, so because then you can no longer trust the findings from the study. So if missingness is substantial, or if missingness is happening more in one arm versus the other, then missingness is really, really concerning. And there are approaches that we will take to address missingness. These are all statistical approaches, and the decision will then be what is best for this particular scenario. So I'm going to, in the next few minutes, and we will wrap up, show you the results from the IVON-PP trial, but I'm going to just show you essentially how some of the decisions that we make might influence the results. Okay, this is the result. This is IVON-PP trial. IVON-PP, if you're not familiar, is a trial of intravenous iron versus oral iron among postpartum women in Nigeria. Okay, so women all over the country, at the time when they deliver within the first 72 hours, we'll provide them with either intravenous iron, or we will provide them with daily oral iron that they will take for 6 weeks. And then we will assess them at the end of the 6 weeks. Okay, and these are the key outcomes. The primary endpoint for the IVON-PP was anemia at 6 weeks. This is a very important question because women who have anemia are more likely to die, right? They're also more likely to experience depression in the first few weeks. That means they're less likely to breastfeed their kids, their babies. That means that the babies are now less likely to grow properly. So you can see how the effects cascade. So this is a very important question that we have assessed, and this is what we found. The pregnant women, the women who use intravenous, who were in intravenous arm, fewer of them had anemia at 6 weeks. So 18% compared to 32% who were on the oral iron arm had anemia at 6 weeks. Okay, and you can see that the risk ratios are less than 1, and they don't cross 1. The confidence intervals don't cross 1. And you see that is consistent for anemia. It's also consistent for moderate to severe anemia. And when we followed the women all the way until 6 months, they also still had that effect. Okay? So this is good, right? But when we then look at the effects on the kids, we found that there is more infant deaths among the kids who receive IV iron. Small numbers, right? Small numbers, but every death in a child is concerning, right? So now we go from 0.6% in those who receive oral iron to 2.4% in those who receive intravenous iron. And this is how a lot of clinical trial results tend to be. You're seeing all these exciting results somewhere, but then there's a safety concern, and that gives you pause. Now you're wondering, where do you draw the line? How do you strike the balance between the efficacy and the safety of the intervention? But I want to call your attention to the footnote. And usually when we're reporting results, we'll report how many people had missing information. And in IVON-PP, we had 46 women in the IV iron and 37 women in the oral iron were missing anemia at 6 weeks. Okay, and if you think about that, that's maybe 7% of the population. So it's not a small proportion of the participants. So there's two questions that you will have when you're looking at missing observation in clinical trials. One is, how large is that missingness? And the second is that missingness the same in both arms, right? So here we are finding that it's not much different in both arms. And I'll talk about that a little bit more as we go. The other thing that you then want to look at is to look at those who actually miss. Look at the people for whom there's missing information, and now you can compare them. Look at who are they? Because sometimes understanding who is missing information can give you some glimpse into why they are missing, but also can reassure you as to what's going on. So in this case, we looked at that. And when we look at the pattern of missingness in IVON-PP versus in IVON-PP, the first thing I want to call your attention to is to compare those who are missing overall, the 83 women who had some missing data, and those who did not have any missing data, right? Those who were the complete cases who had no missing data. And if you look at the educational attainment, you will see that those who had missing data were more likely to have had tertiary education and less likely to have had primary or none, right? So that means that the more educated people are actually the ones that are more likely to be missing. So it might be potentially an example, a possible explanation might be they had to go back to work early, right? And because they had to go back to work early, now they're not available for clinic visits or for you to collect their blood sample. Or, sometimes when people are highly educated, then they're elitist. They're like, oh, I don't want to get involved in all that, or you can't take my blood sample, or maybe they're also more skeptical of clinical trials, right? So understanding that is important. So now we're seeing that this is how the people who have data are different from those who do not. But there's another thing that we also see is that there is more missing data in the rivers versus in all the other states where we had conducted the study. Okay, so now if you were trying to study this in more detail, you will go to the sites in rivers and try to understand why there was more missing data there. We also looked at the people who had iron deficiency, at baseline. And you will see that there is slightly more iron deficiency in those who had complete cases. So it might be those people had less iron deficiency, they were probably healthier, and so they were able to go back to work, and that's the explanation for their missingness. But the other thing that you will look at with missing data is whether the outcome is differentially missing in the study arms, right? So I've earlier said that 46 and 37 is not much difference, right? That's maybe about 9 people difference, so not much. But are the people who had missing data, are they different in those arms? And so if you compare the 46 and 37, you will see that age first, there's older, the people who had missing information in the IV arm we're slightly older, not much difference, but slightly older, 30 and 28. And then if you also look at educational attainment, you will see that those who had missing information in the oral arm were more likely to be younger. So maybe they had less confidence in the oral treatment. If they had received IV, they would have been more like they would have not missed that information. And then now we come back to rivers, you see that the missingness was more in the IV arm, 41% compared to 30%. Okay? So these are ways that we will try to understand the missingness. And so the key question will then be, so what? What should we then do about this missing information? And we will usually conduct sensitivity analysis. The first line here is the main analysis results that we looked at before, okay? We will usually then, one of the things that what we did in I1PP was multiple imputation. So we used multiple imputation to address the missingness in I1PP. And by the time we had used multiple imputation, the results that we got were now very similar. Even after addressing the challenges of the data, you still have findings that are very similar. So which means, in essence, if you did not do anything to this analysis, you would have still been fine. But the fact that you have now addressed it, you feel even more confident, okay? But the other thing that we would usually do is subgroup analysis. And I'll just highlight to you, like if you look at across the different states, right? Across the different states, there is statistically significant effect in all the other states except rivers. Okay? And remember that we saw how those differences in rivers, in the missing information in rivers was important, right? And that carries through to what we are looking at here in our analysis findings. You might argue that maybe this is very close to null. 1.04 is not much different, but the key takeaway for me is, first of all, people benefited across the board, but this missingness in rivers is likely to have impacted the results in rivers as well. So this is one illustration of how the decisions that you make in clinical trials has potential to impact your findings. We've now come to the end of this talk, and these are the key takeaways that I would want you to walk out of this room with. The first is that trustworthy evidence in clinical trials depends on thoughtful design, right? When you're conceiving the clinical trial, you want to ensure that you're anticipating the challenges. You want to go from that target experiment to the real experiment that you can conduct and understand the differences along the path. Randomization and blinding are the bread and butter of clinical trials, but they're not just technical details. They exist because they help us to ensure that clinical trials are fair and that they are rigorous. Data quality, data cleaning, and proper handling of missing values is really, really central to clinical trial. It's not just, what statistical test should I use? Right? You have to think and you can see that throughout this conversation, I have not talked about p-values, I have not talked about regression, I have not talked about chi-square or whatever it is that we're all familiar with. The things that really matter to clinical trials is not just the statistical test. It is also all these decisions that we are making throughout the conduct of the trial that determine whether we can obtain valid and rigorous results. Finally, the population that you include in the clinical trial is really, really central. And if you don't remember anything else from this talk today, you should remember that the intention to treat population is the most unbiased population in clinical trials. But there are other populations that we will be interested to look at to help us to understand the data better, but you must always, always report the intention to treat population. Thank you very much.
Thank you very much, Dr. Abioye. That was very insightful. So we're going to move on straight to the question and answer session, and we'll do this in a very, okay, all right, okay. So we'll also take note of the questions on Zoom. So please, if you have a question, you can just signify so that we can take all the questions, note it down, and then Dr. Abioye can respond to them. So if you have a question, please signify so that we can. Okay, so we have 1, 2. Please raise your hand so that, yeah. So we have 1, 2, 3, 4, 5, 6, 7. That's all. 8. Did I count you? I think I did. Okay. So, Victor, we can start with, yeah.
Thank you, Dr. Abioye, for the insightful presentation. That was great. Just a few comments, and my question will come last. I'm glad you trashed out the issue of the impact of faith in learning on evidence synthesis in clinical trials. Then you also talked about blinding. You then specifically talk about the role of placebo in facilitating blinding, because that also helps to improve the quality of evidence derived from clinical trials. Then for the study design, or rather the trial design, maybe if you can throw a little more light with examples, how trial design can impact the quality of evidence generated. Then sample size, I think you talked about that at some point, the impact of low sample size on the quality of evidence generated. Now, to my question, I think here you talk about allocation concealment, if I'm right. There is something that can also affect the quality of evidence. Then out of visit window. In fact, there's something, you know what I'm talking about. I know in iPhone PP, for instance, we ask out of visit. The question is, how wide should that margin be when you are talking about out of visit window? Because it gets to an extent, it might actually be damaging to the quality of evidence you are generating. When the windows are down to wide, let's say you are to assess something at 10 weeks time point, and then you are now saying, giving an error margin of plus or minus 6 weeks, making it 4 to 16 weeks. That's a very wide margin. Yes, it will reduce your missingness drastically. But what is the quality of evidence we can derive from that? So aside from this, I don't think I have any other questions. Thank you.
Thank you, Dr. Abioye. My question is just a little bit on this. Before I ask the question, I think it's right to appreciate you for that wonderful lecture. I learned a lot, and I hope others also got great feedback. My own is just it's about this DSMB, and of course we need to do interim analysis. I feel sometimes it's decision of DSMB to stop the trial based solely on the interim analysis, or you have to put other things into consideration in terms of whether you want to stop or based on the data or the findings you are getting. And I have a bit of challenge with what the DSMB, decision usually based on the interim analysis. Because I know that, sometimes you make provision for multiple testing. But I just want to know, I usually have the impression that maybe the analysis that based on those probably not able to get feedback because it's really not. Okay, because the study is not really powered to detect effect at that point of interim analysis. So how do we bypass all that? And of course, I also wanted to throw a little bit of light on the concept of counterfactual effect. Maybe just a little bit of understanding would be appreciated. Thank you very much.
Good afternoon, everyone. Thank you, Dr. Abioye, for your seminar. I wanted to ask two questions. The first one is, can you give us some real life examples of protocol deviations and how they have affected the trials you've done? And also, I'm sure it's not a seamless process to enroll Nigerian patients into clinical trials. What obstacles have you faced and how have you been able to overcome them? Thank you.
I want to clarify some things. The first thing is, as regards sample size in clinical trials, what do we consider, or let me put it this way, what are the factors that determine sample size calculation in clinical trial? Then two, protocol deviation and violations. Is it a must? Is it something that is expected? I know you told us that maybe so percentage, but is it possible that we have a clinical trial that we don't have or we must have? I want to know. And the third one is practically, apart from BSMB, TSI, and all those TSC, is it possible as a PI of a clinical trial to see that, oh, this clinical trial is not going on well, I want to retrain and I stop for a while. I just want to know practically.
Absolutely, Dr. Adi. I have a few questions. The first one is, we talked about precision and bias, how important they are in clinical trials. So, my first question is, how do social and economic and cultural biases in Nigeria affect research and clinical trials? And then secondly, you talked about the character that is involved in actionability. So the question here is, how do we validate the adoption of this actionability if you are dealing with human subjects? And lastly, didn't explain how large variability can be a potential source of error in clinical trials. Thank you.
Good afternoon, sir. Thank you very much for such an insightful session. So I wanted to ask about field trials. I wanted to understand how um, you see field trials. How do you detect if the field trial is as a result of errors or as a result of ineffectiveness of the intervention? And then as a researcher, how do you respond to the field trials? And how do you communicate those trials to policymakers and the government? Thank you.
Field trial.
Thank you very much, sir, for the wonderful presentation. It was very succinct and I thoroughly loved it. So I have three questions here. The first question is concerning the DSMB, how often do they review the study? Is it, because I know that clinical trials also come in phases, so is it after you've started a phase or when you finished one phase that they review, or there's a time interval that they do that periodic review to see how beneficial it is to the patient? Then my second question is for the missingness of the in one arm of the study, when you notice that it is very significant and there's no how you can, you cannot just throw away the study like that. Is there a way to equalize it in such a way that the arm that is a lot, you randomize it so that it becomes equal with the other one that there's nothing you can do about, or what do you do about that kind of thing? Then the third one is the triple blinding, the health worker, everybody doesn't know who is who. So I'm wondering how will you, I'm wondering who knows. You understand? Because if you analyze, you need to know, okay, these are the results I'm getting from the people who were with placebo. So how do you handle such in triple blinding? I believe we've taken everybody's
questions in the hall? Okay.
Thank you so much, Dr. Abioye, for the wonderful seminar, well delivered. My question, I don't know if it's beyond the scope of your seminar, but I still want to put it out. It's on budget, costing. What does it cost to run a clinical trial in Nigeria? Also, for the different types of trial, what should we be thinking about in terms of costing? What is the right way to do like a proper costing for a clinical trial? Thank you.
Okay. Yeah, just a follow-up question that I forgot to ask the first. So it's about this composite endpoint. Is on how to calculate sample size for a composite endpoint. So I've really had a lot of challenge with navigating that. So will your sample size calculation, what figure do you put into the calculation? Is it going to be based on individual, your component endpoints? How do you navigate that to make sure that your study is actually adequately powered for composite endpoint? Thank you.
Do you have any? Very much.
All right, sorry, Dr. Abioye, just before you respond, just one more on Zoom. So the question, the person actually has 4 questions. And the first one is, please, where the healthcare worker and outcome assessor, PI, are blinded, but not the participants, is that also a double-blind RCT? Okay, then the second one is, could you please shed more light on modified ITT? Then the third one, could you please summarize how data analysis for ITT and per protocol differ by merely looking at the consult flow chart? I also need clarification on when in the data analysis are participants dropped out of the analysis, when they either did not complete the study due to preterm delivery, miscarriage, or due to dropouts. Then the fourth one, in an RCT, where one of the baseline characteristics, say BMI, after analysis turned out to be skewed at the level of normality testing, is that a difference in the effect of a drug? In that, a difference in the effect of a drug is not found in the 2 groups after analyzing the data. I can show you the questions, but that's all. Yes.
The third question.
Okay, the third question. She says, could you summarize how data
ICT and probable data.
Yeah.
Oh, I see. Okay.
Yeah. Then I think the ending part of that number 4 is, what effect does this have on the results? How could it be interpreted in the discussion? That's the skewed. Yeah. Okay. So, you take the question.
Thank you very much for the questions. This is good, and I'm glad that we are able to ask all these questions. And so I'm going to try to bunch the questions based on the topics and address them to the extent possible. And the first question is relating to blinding. And the first question relates to blinding. And Dr. Baba mentioned that we did not discuss the value of placebo in blinding. Now, a lot of us are familiar with the concept of placebo-controlled trials. And most often when we're blinding randomized controlled trials, we use the placebo. And the placebo, also known as a sham treatment, is basically a treatment that looks exactly like the experimental intervention, that is perceived exactly like the experimental intervention, such that the individual who is interacting with that treatment cannot tell what treatment this is. Okay, that's basically what the placebo is. And so when we say it perceives exactly, so that means the color is the same, the taste is the same, the smell is the same, right? So you will have to, and this is something that is manufacturing, it's a manufacturing challenge. People have to be able to manufacture it properly, and it's not always possible. And so, but most often when you're able to, then you'll be able to achieve blinding, okay? But sometimes you cannot use placebo. If you cannot use placebo, then the experiment, the comparator has to be standard of care. Sometimes, right, you will have to, so I'll give you an example. So say, if the route of administration is the treatment, like in iron, for instance, in Ivan PP, where the route of administration is intravenous versus oral, okay, one question that you will come across in Ivan PP, we had an open label study. The participants could tell whether they received IV or oral. But if you wanted to blind Ivan PP, I wonder if anyone has an idea of how we could have done it. If we wanted Ivan PP to be fully blinded, does anybody have any initial thoughts? Just raise your hand and say, maybe this is what you can do. If we wanted fully blind Ivan PP, we could have provided intravenous intervention to everybody and an oral intervention to everybody. So that means there's going to be two placebos, right? There's a placebo IV and there's placebo oral, right? So that's another way. So placebo is actually very flexible. It allows us to do a lot of things. So it's not necessarily that only one arm has to receive placebo, as long as it helps us to blind. So thank you very much for bringing that up. There was a question about if we provide placebo, right? If we blind the treatment in the triple blind, who will know, right? In all clinical trials, there's always people who know exactly what the person is getting. Okay, there's always people who know. And the person who knows is always the data person. And very often, there is one data person who knows. Okay, I'm looking at Mr. Hamid in the corner, right? And sometimes Mr. Hamid does not know because there is another person who knows, who is removed from the conduct of the study. Okay, and it might be that when I was working, when we're conducting clinical studies in Tanzania and the team at Harvard is involved, there will be someone in Boston who knows exactly what treatment each person is getting, and that person never goes to the field, has no interaction, right? So there's multiple ways that we can ensure that blinding is complete, right? We will separate the person who is aware from the conduct of the study. And there was another question about if the outcome assessor and the healthcare worker are blinded, can we also call that double blind? And my response to that is semantics, right? And that's not really, really important. So the concept of single blind, double blind, and triple blind, these are ways for us they are learning tools. They're ways for us to understand blinding. It doesn't mean that these are the only ways that you must blind. It really depends for each clinical study. It depends on what the design of the clinical study is. Sometimes the patient will not know, but the healthcare worker knows, and the outcome assessor does not know. So an example might be surgery, right? This clinical trials where the intervention is a type of surgery. And you might say maybe this person gets one technique and the other person gets another technique, and you're comparing two techniques. Both patients are wheeled into the theater. They don't know what they're getting, whether they're getting this technique or the other, right? The healthcare worker has to know because they're the one that's conducting the study, right? They're the ones delivering the intervention. But the outcome assessor who is checking maybe bleeding after bleeding the post-op period does not need to know what intervention the person got. Do you see what I mean? So it's the semantics are not really important. They're ways for us to understand how blinding happens. But it doesn't mean that these are the only ways that we can or should blind. We should look at the design of the trial, look at where it's feasible to blind and blind as much as we can. right? So the fact that we cannot blind a healthcare worker does not mean we should not blind the outcome assessor. So we'll blind wherever possible, and I think that's the step that we should take. Sorry, my computer is signed out, and now I cannot remember. So that's blinding. We also talked about, so there's a question about sample size. So sample size is really, really important. And sample size affects precision. Okay? Sample size affects precision. If the sample size of your study is small, is too small, you will not be able to find precise effects. Okay? So what we are hoping to do with increasing the sample size is to improve the precision with which analysis is done. Okay? We talked about allocation concealment, and that's really, so if you remember from my talk, I was talking about how when you generate the allocation sequence and say, if this person comes down, they're going to get treatment A, the next person gets treatment B, the next person gets treatment B, then the next person gets A, then B, then B, then B, then A, you know, like, so you're going to randomize. You're not the one who is choosing, the computer is choosing for you. And that allocation sequence, you do not share it to whoever does not need to know. Okay? You do not share it to whoever does not need to know. And you only share it at the point when they need to know. So if, for instance, let's talk about this surgical trial that I was just saying. If it's a technique that the surgeon is very comfortable with, you don't tell that surgeon until it's time for them to go into the theater. So as they're going to the theater, the patient is already in and prepared, or maybe when the surgeon is at the bedside, then they will say, okay, what surgery is this person getting, what technique, and so they're getting this technique. That's the point when the surgeon has to be aware, right? So allocation concealment, it really also depends on when the person needs to become aware of it. So it's part of blinding, right? Because it helps to keep the blinding intact. There was a question about variability and out of window visits. How wide should the window be? And there's another related question. I'm trying to find it. So the first question in this block is, how wide should the window be? And the reality is that it's a judgment call. And that judgment depends on our understanding of the biology. Right? So if we expect, for instance, that hemoglobin does not change meaningfully over a couple of weeks, right? That red blood cells take 90 days to be, so that means you have a little bit of leeway. If you assess around that 90 days, a week before, a week after, you're still assessing the same hemoglobin imprecisely. Okay? So how should you determine how wide the window is? It's not a statistical question. It's really a clinical question. It's a question that depends on your understanding of the subject matter. So that's something that we should bear in mind. But if the window is too wide, then that's a statistical question, because then you have imprecise estimates, right? You have imprecise estimates, and what should you do if you have imprecise estimates? One way that you can get around imprecision beyond actually assessing better, ideally the way to get around it is to design better, to ensure that you're assessing your measures are as accurate and as precise as possible. But the larger the sample size that you have, then sample size helps you to address precision to some extent. A large sample size helps you to address precision to some extent. Let's talk about enrollment, okay, enrollment. And there was a question about what are the unique challenges in the Nigerian setting, okay? One thing that I want to tell you is that we like to think, and this is a belief that I have, and this is not clinical trials, we like to think that Nigerians are a special group of people, that they're atypical, maybe they fell out from heaven or something, and they're completely different from the rest of the world. This is not true, okay? This This is not true. We have to disabuse our minds. Every setting has unique challenges that make them a bit different, but there's always challenges everywhere in the world, right? Every community where you conduct a study have their own unique challenges, okay? If you're conducting studies in the US, for instance, there's a lot of people in the US who who are uncertain about clinical trials. They think that maybe the drug industry is trying to poison them, and they have all these views, okay? And that in Nigeria as well, people have concerns about clinical trials that they might impact the extent to which they will participate. And the most likely concern that people will have is whether they have time for it, right? Whether they can come to the clinic. So that's an important challenge. But what I will say the most important challenge that we've experienced is with consenting, right? A lot of the studies that we've conducted at CCTRIS are among women. And in Nigeria, when you consent a married woman, you ask a married woman whether she wants to participate, it is very likely that that decision is not made by her alone, okay? Even when you consent and she agrees and says, oh yes, I'm going to participate, you should assume at the back of your mind that the next time that you interact with her is when you will know for sure whether she truly wants to participate. Because then she's going to go home and ask her husband and discuss with him, and then they will come to an agreement and say, and if he says, oh, why would you do that, blah, 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 trial over, right? And we have a lot of cases where a woman then calls back a few days later and withdraws. Or you call her and now the phone is not getting through. Eventually you find out that her husband did not consent. This is not a Nigerian problem. It's everywhere, right? I've led clinical trials in Tanzania. We have this exact same problem. In fact, what we would usually do is that you will provide the consent form to the woman so she can take home. And then when she comes back the next week, so the screening visit will happen first. You tell her about the study, she's eligible, and then you give her the consent form so she can take it home. And then when she comes back the next week, so you will schedule another visit and say, oh, can you come back in a week's time? That's additional costs to the trial. But that's when you can actually be sure that the person who does come back actually consented to your study, right? So this is an issue that we experience. It's not just Nigerian, and it's also related to our socioeconomic and cultural biases, especially in African settings, or in settings where the influence of the family unit tends to be much greater than any single individual, and especially in settings where you have men who are not engaging with the healthcare system as much as possible. And this is the reality. Like, if you try to conduct a trial among adult men today in Nigeria, the thing that you will experience it's going to be very challenging, and this is the truth, unfortunately. But these are the issues, and like I said, there are challenges everywhere in the world, and understanding and anticipating them and designing your study to address and prevent them is critical. So let's talk about the DSMB. Okay, the DSMB, there was a question about interim analysis, what considerations so interim analysis, so when you set up the DSMB, remember I said there's a group of independent people, usually professors, who, when they're starting a trial, you ask them, can you please help us with the DSMB for this study? And then they will agree. The DSMB has to meet before the trial starts, during the course of the trial, and best practice at the end of the trial. Okay? Now, during the course of the trial, depending on how long the trial is, a good duration of time between meetings is 6 months. Okay, so you want the DSMB to meet as often as possible, because if the DSMB meetings are too far apart, then it's not likely that they will benefit from, these people will forget the details of the trial, so if you're 1 year apart, it's so long, it's so far away. But 6 months helps. They can actually truly provide the best insights to you. That's how far apart the DSMB should happen. And there was a question of the interim analysis. So every time the DSMB meets, the DSMB will look at the data that you have already collected in the study. The first time they will look at the protocol and align with you on how the monitoring is going to happen. And then every time that they meet thereafter, they will look at the data. Okay? And usually, like remember that I said that the first thing that DSMB cares about is safety. So usually at every visit, the DSMB will look at the safety outcomes. Look at what the adverse events that patients are experiencing, make sure that they're not too much, and make sure that they are not happening too much in one arm. But there's the interim analysis that is then sometimes part of the trial. And the first question about the interim analysis, this is the time when the DSMB looks at the full spectrum of the data. And sometimes the DSMB will, rather than look at all the endpoints, they would only look at the primary endpoint. Okay, but the key thing is that the DSMB will look at that by study arm and then use that to make a decision. I would say that in terms of best practice, the DSMB should be focusing on the primary endpoint as well as all the safety endpoints. At what point should the DSMB do this? At least when you have 50% of the population enrolled. Okay, so once you've enrolled 50%, that's enough for you to get a glimpse. The challenge is that if you have done your sample size estimation and you expected that 1,000 people will be required for you to detect the effect, right? Target sample size is 1,000. Now at 500, you're doing the interim analysis. You really do not have enough power to detect an effect. But you have to, but what the interim analysis therefore does is not to fully and completely say that this is the finding of the trial. It is to give you a guidance, and that's why people are moving away from saying stopping rules. Instead, we're saying stopping guidelines, right? Because then you will look at like I said, the safety outcomes and see whether it's more in one arm. But you would also look at the primary endpoints and you would use statistical guidelines to determine whether the DSMB should consider stopping the study. And if the DSMB finds something that it finds concerning and wants to stop the study, it needs to take a pause. So let's say, for instance, in this trial, you find overwhelming benefits in the trial. All of a sudden, you're seeing that all these people are having, this anemia is so low in this group versus the other. And, like an Ivan PP, for instance, I showed you the result. So if the DSMB had seen these results early in the trial, they have to make a decision. They have to ask themselves, given all the missing data that exists in this trial, right? Because usually when you're conducting the analysis, when you're conducting a trial, there's often some missing data at the time of the DSMB because you haven't finished. So the DSMB has to consider, given the missing data that exists, will the result change? So if, for instance, 20% was missing data, if all those 20% did have data, what could the result have been? So that's one. The other key question that DSMB will consider is long-term. So say, for instance, the majority of the participants have only been in the study for, let's say, 2 weeks, but the outcome of interest is 6 weeks. The DSMB has to compare that 2 weeks data with the 6 weeks and make sure that there's an actual consistent trend in the results. And because the challenge is, if you look at the result now and it looks like it's looking okay, but what happens over time, right, as more people are enrolling? So although those stopping rules exist or stopping guidelines, the DSMB should not see them and just jump at them and say, oh, now they have to sit back and ask themselves and have some degree of judgment about this and say, is there any chance that this data is going to remain the way it is, or is it going to switch? So that's the thing about the DSMB and interim analysis. Sorry. Dr. Kwande asked about the counterfactual. Okay, this is a key concept in epidemiology that carries over into clinical trials. And in reality, what we're thinking about in the counterfactual is also exchangeability. Okay, now I like exchangeability because it's more explainable. The terminology actually explains itself. When we're thinking about the counterfactual, what we're really saying is, if you look at the findings as they are today, or if you look at this scenario as they are today, in a different world, you imagine a different world, and that had a different set of inputs, what will the result have been? I think this is quite philosophical, and it's hard to explain the counterfactual without being a bit philosophical. But if you remember, I gave the example that when a patient walks into your clinic, if you're able to duplicate that patient, and for one copy of the patient, you gave them the treatment, and another copy of the patient, you did not give them the treatment, and then look at the effect. Basically, what you're doing there is that you're creating a counterfactual of that patient. It's a philosophical idea that helps you to understand what we're trying to achieve in clinical trials, right? Now, it's also an idea of what we're trying to achieve in epidemiologic studies in general with limitations, right? So it's just helpful for you to kind of understand it as this is the goal. One question that we got was, how do you validate the exchangeability? And the way that we would usually do that is by looking at the baseline characteristics. So you look at the table of baseline characteristics, and you will look to see whether the people in general look similar. There is one debate in looking at the table 1 of clinical trials where baseline characteristics are presented. Some people will say that you should present the p-values so that you can statistically say that maybe the age is different, maybe the gender is different, but some others will say that maybe you don't need to present the p-values and you can just look at the results. And because the issue is that what we're looking for is not a perfect comparison. What we're looking for is a general trend. We want to see whether people are roughly similar to each other. Okay, and if we are randomized, then the results should hold correct. There was a question about protocol deviations and violations, and whether it is possible to have a clinical trial without protocol deviations and violations. I don't think it's possible. Okay, I don't think it's possible. I think that human beings are human beings, right? And as a clinical trial, initially, so for instance, as you're starting a clinical trial, people have just been trained on something, and now they're having to work with that protocol. There's a group of protocol deviations that are likely to happen at that time that come from the fact that people have not truly learned the protocol properly, right? So that's one group. Another challenge then is, over time, people get accustomed to doing things in some way, and as a result, they don't fully apply to the settings where it should not apply. So, for instance, eligibility. When you're starting a clinical trial, when people are still learning the eligibility criteria properly, it is very likely that 1 or 2 patients, right, will be enrolled into the clinical trial that should not be there. That's a protocol violation, or also known as a major protocol deviation. So that's a challenge that you want to avoid, but it's hard. So that's why we also then have processes to do per protocol analysis where you can actually compare those that don't have the deviations to those who do. So is it possible to not have deviations? I think it's very difficult. It's possible, but it's very difficult because we always have human beings that are conducting the studies. Can a PI stop a trial due to concerns about conduct? Yes. Yes. A PI can stop a trial due to concerns about conduct, but it is not advisable. Okay? It is not advisable. The reason why it's not advisable is because a single individual whose career, for instance, is linked to the success of the trial, right, is very likely to be biased. You might think that you're looking at a trial rigorously and you're doing your best in being objective, but because you have a conflict of interest, and you do have a conflict of interest, you want the trial to be successful, right? So it is best that you bring your case to a DSMB and let this group of independent adjudicators help you sort it out and say, oh, we have these concerns, as opposed to you then deciding that we want to stop this trial now, because the truth is that you're not likely to be objective or fully objective. And even if you were objective, the person who is looking at the situation might not believe that you are truly objective. So it's both what you think that you're doing, but also what others are perceiving as well when they look at the scenario. There's a question about how we respond to failed trials. Is it always an error in implementation, or should we just believe that the intervention is ineffective? And the truth is that it really depends on a lot of factors. Okay, it depends on a lot of factors. And one of those factors that lead to failure of trials is the design. Failure of trials is a huge issue. Every year, the drug industry spends billions of dollars trying to bring new treatments, right, to the market, and many of these drugs fail in the pipeline. So it's not a small issue, and that failure, sometimes it happens because you have not selected the right group of patients. Sometimes it happens because the dose of the intervention is wrong, right? And then it's not effective amongst them. Sometimes, and going back to the right group of patients, to give you a little bit more detail, an example is, I don't know if anybody here followed the controversy of aducanumab, which was an Alzheimer's drug that was developed a couple years ago, I think maybe 3 years ago. And when you looked at the clinical trial results, the primary endpoint was not met, but the trial was effective for other things. And so the company was like, oh, what can they do about this? But really what happened with that trial was that the selection of the patients was wrong. So sometimes when you're looking at, especially like a chronic illness like that, you either want to enroll people who are early on in the disease, where the biology actually should drive the decision. You want to make sure that it's a period when the intervention is actually effective in them, and not just selecting it because of maybe market forces. And, drug trials, pharmaceutical companies want the group of patients that they can treat with their drug to be huge. But if they select the wrong group of patients, they might end up wasting their money. So there's multiple reasons. It might be that the drug or the intervention is not effective in that group of patients, and there's a different group of patients in whom it is effective. It might be that the trial was not properly implemented, but that's not as common, right? It's more common that actually the design is faulty. Sometimes the analysis is the challenge. Where, they have not analyzed in the right subgroups. Because sometimes a clinical trial might not be effective, might not find a good result, or might not find a positive result in the full population. But if you analyze the subgroups, you might realize that maybe among those who have a high BMI, this drug is effective. And that gives you a place to start for further inquiry, even when it's not effective in the population that you have studied. So I think I will try to wrap up. while answering maybe two more questions. And one of them is missingness. What can we do about if there is missingness in one arm? There are statistical approaches to address missingness in one arm. Those statistical approaches will not fully address it, but they will give you a comparison, something to compare with. And if you look at the analysis that we did earlier, that I showed you earlier, we use multiple imputation. Right? There are other ways. There's IPW weighting, which is inverse probability weighting, and there's other approaches as well. There was a question about MITT, and I will take that question and then the budget, and we'll stop. So MITT, we said that the ITT population is the one that is unbiased. But sometimes the ITT is in intention to treat, but sometimes people want to use a modified intention to treat. And basically, the difference between the ITT and the MITT is that in the MITT, you would select a group of people who are most likely to have the outcome. So I'll give you an example. In Ivan is the trial of intravenous and versus oral iron in pregnant women. The intention to treat was everybody who was randomly assigned to which therapy, either group. But we selected a modified intention to treat population as those people who had the week 36 visit. Right? So the woman is already pregnant and she's continuing. And then at week 36, we collect samples. That's when we collect the data on the primary endpoint. So one way is to select those who are most likely to have the outcome. Another way is to select those who are most likely to have received the treatment, and this is one most common. So for instance, if this is an intervention that people are collecting over a long period of time, you might say the people who got at least one dose of the drug, those will be the MITT. And you see, and as you can see, different ways that I'm describing it, the question of who becomes included in the MITT depends on what the investigators choose. Okay, there is no standard per se. And because it depends on what the investigators choose, is one of the reasons why the MITT, although it is highly regarded, is less highly regarded as the ITT. Okay, because there's a little bit of judgment that's involved. The power protocol population tends to be the group of people who actually followed the protocol, right? Those who did not have major protocol deviations, those will be the power protocol. And as you can imagine, it means that you've excluded all these people who you're excluding for a reason that is related, that might be related to the outcome, right? So that makes the power protocol analysis even less attractive. And the questioner asked, when you think about the consort flow chart, at what point would you exclude those who will not be in the MITT? And it really depends on what the investigators choose to do, right? If you have specific questions about this, I'm happy to discuss later about, with your questions. Final question is probably the most important question, and that's the budget. We like to think about clinical trials, but they are very expensive ventures. Or let me say that they can be expensive ventures, but they don't have to be. Okay, they don't have to be. The key thing that you need to think about is, what is the target experiment that I want to conduct? What resources do I have? To what extent, what can I really do using the resources that I have? And then design your clinical trial that is as rigorous as possible, but bearing in mind the limited resources. So an example will be, for instance, instead of providing the intervention, are these interventions that the patients would already buy? So that's one thing to think about. But then you have to think about the potential bias from that. instead of being the one who pays for the outcome assessment, say an example, MRI, right? You're conducting a study and the MRI is maybe the imaging outcome, say it's a neurologic trial. Will this be a trial that the patients will already pay for the MRI? So the way to think about the cost is to ask yourself, what are the things I can get away with while preserving the rigor, right? Another reason why cost is, why clinical trials can be expensive is long follow-up times, and that large number of staff, right? Can you cut back on that? So those are ways that you can modify and keep a clinical trial as cheap as possible. And it really depends on the study. So you can have one research staff for a clinical trial if it's just, say, the surgical trial that I was talking about, where there's just one research staff, and that's a person who knows the intervention and who tells the surgeon the intervention, which particular group the person is. Like, it doesn't have to be huge Ivan PP, right? It doesn't have to be A's and V. That's the point I'm trying to make. You should take a look at it from the specifics of the study. And how would you calculate the budget? You really should look at the elements of the clinical trial, right? If you're providing the intervention, what does the intervention cost? If you're paying for staff, what do those cost? And those are often the two most important, those are often the two most expensive budget items in clinical trials. Another one that might be expensive, depending on what it is the assessment. So if you're paying for MRIs, if you're conducting blood tests that are not regularly available, they're not routine, then that might be another area where the clinical trial then quickly becomes expensive. But in budgeting for a clinical trial, you separate it into its parts. And it's really the same way that you will budget for anything that you're trying to do. You're trying to build a house, you're asking yourself, how much cement do I need? How much do I need to pay my laborers? So it's really, the budgeting piece of a clinical trial is not a statistical issue. It is a project management issue. So you break down into the elements and cost it and figure out and project and have extra room on the side so that if your budget is not perfect, you can have at least some breathing room, miscellaneous and the like. And yeah, so that's really how you will determine the budget for a clinical trial. Thank you very much. Thank you very much for all the questions. I appreciate them.
Thank you very much, Dr. Abioye. Sure. One very kindly question. It was on Zoom and it's about Ivan PP trial, talking about attrition rate.
So the question is, for the Ivan PP trial, was there an estimated attrition rate? What was the rate? And does controlling for attrition have any effect on data imputation method for controlling for missingness? What happens when you're unable to get the required sample size, especially when you cannot get the required disease patients? Part of this question is in the design and part of it is in the analysis. So was there an estimated attrition rate? Yes, so there should always be an estimated attrition rate. So you would ask yourself, based on the trials that we've conducted, what group of patients do we lose? We usually use 10 or 20%, and we would estimate that, we want a large enough sample size that even if 10% of the population or 20% of the population do not show up or are lost to follow up, we'll still have a large enough sample size to detect an effect. Should the estimated attrition rate affect your imputation? No. It should not affect your imputation, right? It should not affect the approach that you use to address the missing data. Now, this is different from the actual attrition that you observe. If you have a huge attrition, but you don't have a lot of missingness anyway, right? So the outcomes are complete except for those who were lost to follow-up, for instance, then you might make a decision about the analysis approach based on on the extent of attrition. You might say, oh, maybe because we only want to adjust for attrition, then we can use maybe in IP weighting. But or if you think that there's multiple different ways that you will, multiple different factors that might be responsible, and the missingness mechanism, and this is a term that I'm trying to avoid because it's fairly technical, but the way in which the missingness is, what you expect of the missingness. Do you think that the missingness is at random? I.e., there's no way that you can predict. It just happened that 50% of the population could not, which is unlikely because that's such huge. Or it just happened that 5% of the population don't have the outcome information, right? And it's random. Because it's random, you will feel comfortable using certain particular analysis approaches. Or if you think that the missingness was not at random, then you're really in trouble because there's not very good approaches that can address the missingness, right? So it's important to understand the approach, the mechanism by which the missingness is happening, because then that will help you guide the analysis approach. But it does not really depend on attrition alone. It depends on multiple factors, and attrition is just one of the factors. Thank you.
Thank you so much, Dr. Abioye. Thank you for answering all the questions, and thank you for that insightful lecture. We are very grateful. So we've come to the end, and just before we say our goodbyes and network, we have the closing remarks from our phase 3 clinical trial lead, and that is Associate Prof. Baba.
Thank you so much for coming here to make it clear to us that good evidence synthesis starts from the design of the protocol itself. I don't know what lesson you are taking home, but I'm taking home 3 key lessons from what Dr. Abioye has been telling us. 1, that in designing the protocol, I need to be crystal clear. 2, in data management and analysis, I need to be transparent. And 3, in making interpretation and conclusion, I need to be unbiased. That is the only way we can help the world. And that's what CCTRIS is trying to push out there, to train researchers, to make you develop skills, to make you know the do's and don'ts in conducting research, so that from our beloved country, Nigeria, we can produce good evidence to change the world as far as clinical practice is concerned. Once again, thank you so much, Dr. Ibraheem Abioye, for coming all the way down to teach us. And to everyone present here, physically and online, I want to sincerely appreciate you all for making out time to be with us. Then I want to use this forum to disseminate this information to you that CCTRIS is on YouTube, and the webinars are usually posted. So we shall be sharing this on the various WhatsApp group. You can visit the CCTRIS website for more, and you can also watch webinars that you are interested in. Thank you all for coming.